Alright, so we're going to start talking about evolution and the history of life. So we'll be talking about all of this, how this whole process occurred and how the history of life developed over time. So, evolution, the process that has transformed life on Earth from its earliest forms to the vast diversity that we have characterized today. And it's basically a change in the genes of those organisms. So we get this change of genes that affects how the organism is able to adapt to its environment, how it reacts in its environment, all that kind of great stuff. So the old theories of evolution, first one, or one of the first ones, was Jean-Baptiste Lamarck. This is the one that you got to know, other than, of course, our Charles Darwin. He was in the early 1800s, and he proposed the idea of the inheritance of acquired characteristics. There are a lot of people that had different ideas about evolution. Charles Darwin didn't come up with the idea of evolution. He came up with how evolution occurred. And Jean Lamarck also did the same thing. So his idea was this idea that as organisms use these different characteristics, they're able to adapt those characteristics during their lifetime, and then be able to pass those along to their offspring. And his great example was the giraffe. Right? So the giraffe acquired its long neck because its ancestors stretched higher and higher into the trees, reaching up higher, causing their necks to stretch, and then they were able to pass those around to their next offspring, and then those offspring stretched more, making their necks longer, and then they passed those along, and then over generation to generation, the giraffe's neck slowly got longer and longer and longer. And so that was Jean-Baptiste Lamarck's idea. Of course, we know now that with basic genetics, that this is not how this process typically occurred. But if you also remember back to 101 a little bit, if you learned epigenetics, epigenetics is that idea of how actually some things in your life, in your lifetime, can actually affect how your DNA is expressed. It doesn't change the A's, T's, C's, or G's, but it can affect how what genes are turned on and what genes are turned off. And so this actually is not completely off the mark in that idea. So they're starting to look back at Lamarck's ideas and Lamarck's propositions, and he's getting a little bit more credit. But at the time, it was kind of poo-pooed and looked down upon. Then, of course, we come to our guy named Charles Darwin. So this is the guy you got to know. Darwin ended up working with a guy named Charles Lyell. Charles Lyell started looking at a bunch of data that he had collected while Charles Darwin was also looking at his data. And basically at the same time, they were both kind of coming up with the same idea of natural selection. Little did Charles Lyell know, he kind of sent this letter over to Charles Darwin saying, hey, I've kind of come up with this idea. I know that you've been working on evolution. What do you think about it? And this kind of worried Charles Darwin a little bit because it was the same idea that he had, supposedly. And so he hurried up and published his paper right after receiving this letter. There's been this also kind of overshadow and big controversy with this on whether Charles Darwin actually had the idea of natural selection or whether he stole it from the letter that Charles Lyell sent him. So there's this kind of mystery that goes along in this kind of dark spot that a lot of people don't know about with Charles Darwin as to who actually came up with the idea of natural selection first. But he was the first one to actually publish it and got the credit for it. And what he did is he went out on the HMS Beagle basically to collect species. His job at this point in time was to sail around in the South Seas, all through the Pacific, collecting species and taking it back to England to identify them. So he was basically on a scavenger hunt for new species. And he stopped over in the Galapagos Islands, all kinds of new species there, collected them, had no idea what he was getting into when he was there. So he's not thinking about evolution, he's not having any ideas when it's not like he stepped on the Galapagos Islands and goes, evolution. He just collected animals and then took them back to England to categorize and study. And so he ended up sending off a bunch of different species. He didn't know a whole lot about birds, so he sent them off to a friend who was an ornithologist who studied birds. And his friend worked on them for a couple of months and sent back a letter to Charles Darwin saying that all the birds were actually types of finches. They were new species of finches, but they are all, every single bird that he collected on the Galapagos Islands were all basically types of finches. And so this is what got Charles Darwin thinking, how could every single bird on these islands all be closely related and all be finches? Even though they are feeding differently, they look completely different. You have ones that are eating bugs, ones that are eating cacti, ones that are eating small mammals, ones that are eating seeds, all kinds of different ones, yet they're all basic finches. And so this is what got him to start thinking about his idea, and whether Charles Lyell sent in the information that kind of helped him out with it, or whether he already had the idea and it just spurred him on, we'll never really know. But he ended up writing On the Origin of Species by Means of Natural Selection. What this book did, or what this paper did, is it confirmed or gave the idea that species do evolve over time, and that the mechanism for this is natural selection. Now, natural selection is that individuals with favorable traits are more likely to leave more offspring better suited for their environment. This is the key thing. If you don't take anything away from all these notes that I'm getting ready to go over, the key part is to understand what natural selection is. You need to understand that if you're better suited for your environment, you're going to have some organisms that are better suited, some that are less. You're going to have some organisms that are faster, some that are slower. And if faster is a good thing, then they'll be able to get more food, be able to survive better, escape predators better, and then therefore also be able to produce more offspring. All right? And by making more offspring, the next generation will hopefully carry on those same characteristics as long as they're able to be passed along. And so those traits will be passed along to their offspring, and so the overall population starts to gain those better traits, and the whole population starts to become better and better suited, become faster and faster, or whatever that particular traits are best for the environment, and the whole population starts to become better adapted. And that's the idea of natural selection, and this idea that leads to eventually to evolution. Alright, so it's also known as differential reproduction. You got the ones that can make more babies, some of them can't make as many babies, and so over generations the population slowly becomes more and more like the ones that have more babies. So the big example and kind of the poster child for this idea of natural selection is the English peppered moth. And what happened is back in England, there was this moth species that was in a light color, and it was a light color because all the trees that it lived in the forest that it lived in, the area, were ash trees, and their bark is a very light kind of gray color. And so the moths were this same light gray color with little black dots on them, and it blended in perfectly with the bark of the ash trees. And so the birds couldn't see them very well, so therefore they survived the best. There also within this population was a dark face, but it stood out much more so, and so therefore kind of got picked off pretty easily, kind of like an albino for us, all right, for a lot of species around here. And so this dark face was very rare within the populations. And so what happened is about this time that Charles Darwin came up with this idea of natural selection, they were studying this peppered moth, and we had the Industrial Revolution occur. And during the Industrial Revolution, you had lots of burning of coal and oil and all kinds of different ironworks and productions, and there was tons and tons of soot and acid rain and all kinds of nasty things that fell down all throughout Great Britain where these pepper moths live. And this soot and ash and acid rain caused the ash tree's bark to darken. And so it was no longer this light gray color, but actually turned into a much darker gray color. And so those light phase moths ended up standing out really brightly and got picked off, and the couple of dark phase moths actually blended in very well. And so the entire population shifted very quickly from being 99% light phase moths and only 1% dark phase to the exact opposite, to being almost all dark phase and very few light phase. And we were actually, with somebody studying, what actually followed the entire transition throughout this process, and so we have data going from when they were all light phase to data when they're almost all dark phase. And so we got to see this whole process of natural selection and evolution occur right in front of people, and we have data to kind of back it up. So this was kind of the poster child for this idea of natural selection. Now, we also have a process known as artificial selection, and this is what we do with selective breeding. Great best example is dogs. I mean, look at our common species, or our common dog species, and the differences that we can have within that one species, of going from Great Danes and Irish Wolfhounds that are several hundred pounds down to the tiny little Chihuahuas that you want to boot across the floor that are about 2 pounds. All right, so you have this huge range of organisms all within occurring within just a couple thousand years, and so you get this massive differential, and that's because of the idea of selective breeding. Rather than nature being natural selection, nature being the selecting force, it's us. We're artificial selecting who's going to make the babies and who's not. And we're able to do this much faster. We're not, basically with this process, we don't allow any of the species or any of the traits that we don't want to be bred, and the ones that have all the traits that we want to be bred, they're the ones that have basically all the babies. So the entire population changes very, very quickly. And so we get these massive changes in populations, massive changes within the species very, very quickly, much faster than occurs or naturally through natural selection. And so the idea is the difference between the wolf and the going from a wolf to the common dog species. So natural selection is governed by the principles of genetics. It still happens within there. You still have dominant recessive traits. All those things kind of follow through, and the whole process of how genetics get passed on to the offspring. So those are still big players into how this natural selection process occurs. And we have different types of adaptations. We have protective coloring, like camouflage and mimicry, where like they're pretending to be a poisonous snake or a poisonous butterfly. Camouflage blending into the environment. You can have physiological adaptations, like reproductive changes, like breeding, producing more offspring or less offspring, being able to run faster, being able to digest different foods better, and then behavioral adaptations, like instincts, and like if you ever, one of my favorite examples is tiny little chameleons. As soon as they come out of a shell, they start rocking back and forth and mimicking the leaves around them, kind of waving in the wind. And this is a behavioral trait that's passed on, and an instinct that's passed on to them and it starts working as soon as they pop out of the egg. So it's pretty cool. When we talk about populations, this is what ends up going under evolution, right? Individuals do not evolve. When they talk about evolution, evolution is entire populations changing over time. Individuals do something called mutating. So when we talk about an individual, we get a mutation, right? When that mutation is passed along and starts permeating throughout the entire population, that starts becoming more and more of the population, that's evolution, right? So populations evolve, individuals mutate. So make sure you know the difference between those two. Population produce, what happens with evolution is populations produce more offspring than the environment can support. So you're going to have more organisms out there than can possibly survive. Some of them have to die, some of them will survive and reproduce. Right? So the unequal ability of those individuals, the ones that can get more food are going to be the ones who can survive. The ones who have trouble are the ones who aren't going to make it. And so the ones who have the better abilities, the ones who are able to get the most food, avoid the most predators, and are going to be the ones who can reproduce the most and produce the most offspring, and therefore their genes are going to get passed on to that next generation. The ones who can't do those and the ones who die, obviously their genes are not getting passed along. So the next generation will have more of those genes from the healthier, better adapted parents, and therefore that generation will be better adapted in general. And then the next generation will be even better, and the next generation even better. And so this is how evolution works over many, many generations. And so basically the idea is that animals who have greater fitness survive in their environment and live to reproduce. So if you're better fit for your environment, if you have better traits for your environment, you're going to be the one who reproduces the most and therefore pass along your genes the most often. And random changes, known as mutations, can lead to this greater fitness. Right? And this greater fitness, the mutations that actually increase the greater fitness are called adaptations. So adaptations allow an organism to survive better. So the idea here is that adaptations are mutations that increase fitness. Put them all together there. So adaptations are mutations that increase fitness. They make it so that the organism is better able to survive and reproduce. All right, now we get to the tricky stuff. All right, so with population genetics, all right, so a population again is a group of individuals that belong to the same species in an area. So we can say like the population of raccoons here in Fredericksburg. All right, so that is a population. Right, they're all within the same species and they're in that same area. They're able to interbreed, that kind of thing. So that's what we're talking about when we talk about a population. Now, species is a group of populations whose individuals are able to interbreed and they're able to produce viable offspring. This is the key part. Make sure you highlight and underline viable there. Viable means that that those offspring are able to reproduce on their own also. So the great example here is a horse and a donkey creating a mule. Right? A horse is one species, a donkey is one species. So theoretically, they shouldn't be able to reproduce if because they're different species. But they're able to produce a mule. But the key part here is that mules are not fertile. Mules cannot reproduce. You cannot reproduce a mule with a mule. You can't do a mule with a horse or a mule with a donkey. All right, so mules are not viable, and so therefore that you still are able to have the species of horse and donkey because the mules are not viable offspring. So they don't mess with that idea of what a species is. So the population must be of a species must be able to interbreed, and then their babies must be able to reproduce also. And that's the key to creating to the definition of the species. Now this kind of gets wishy-washy now, especially when you start thinking about bacteria who never have sex, and so how do you classify if they're able to interbreed or not? And we also know species, if you ever looked up liger on the internet, you'll be able to see that you can have a lion and a tiger reproduce, and their offspring are actually do quite well and able to reproduce on their own too. So this idea doesn't quite stand up as much anymore as it did back when Charles Darwin is, but it's still a good basis and a good idea, kind of general definition for a species. And when we talk about a gene pool, we're talking about all the genes possible to that particular population. Now, certain populations might have different gene pools. You might have a population on the East Coast and a population on the West Coast, and they might have very different genes that are available to them. All right? But that gene pool is that collection of genes available to that one population at a particular time. All right. Now, for the good stuff. So when we start talking about populations and start talking about evolution within these populations, we start looking at the genes within those populations, and we try to follow those genes and see which ones are being affected by nature. And we do this, scientists do this by two different methods. They can do it by genotype frequencies and looking by allelic frequencies. So basically we're looking at how the genotypes are in there, big A, little a, little a, that type of thing, how those frequencies are changing over time. And then we also just look at the alleles. How many big A's are in the entire population and how many little a's are in the entire population. Right? So, but we can look at these two differences and kind of see over time if there are changes, or if there's if these frequencies are changing, how those are affecting the population, and start trying to figure out why those frequencies are changing over time. It's how this natural selection is occurring for this population. All right. So the example I'm going to give you are tulips to make it nice and easy. We got white tulips, we got pink tulips, and we got red tulips. All right. White is represented by little r, pink, big R, and red, big R, big R. Okay. Now, for genotypic frequencies, we're going to look at the genotypes. We're going to look at the letters. All right. And so we have a total of 1,000 flowers in this example to make it nice and easy for the math. And so for genotype frequencies, we have 200 little r's. You take the 200 and you divide by the total number of flowers, and that gives us a frequency of 0.2, or 20%. If you're looking at pink, that's a big R, little r. That's 500 of those, so that's half, so 0.5. And so the frequency is 0.5 or 50%. And then red is 300, which is 0.3 or 30%. And so those are our genotype frequencies. And what you can do is over time, you keep taking these frequencies, you keep looking at it, and if these numbers start to change, you can start to follow how the general population might be evolving and changing and try to figure out why that's happening. Allelic frequencies is just simply looking at the letters, the big R and the little r. Right? So with the little r, since they have two little r's, that means we're going to double that number because we're looking at how many of these little r's there are within this entire group. Right? So we have 200 little r's, and that means there's 200 little r here, 200 little r's here, which gives us a total of 400 little r's. If we look down at our pinks, we got a little r in there. So every single one of those has one little r, and so we're going to have 500 little r's within that group. And we're not going to have any little r's within the big R group, right, within the reds. All right, so that means we're going to have a total of 900 little r's within this entire population. All right, as far as big R's go, we'll also have 500 big R's within this pink group because every single one of these guys has one big R, so there's 500 of those. Within our red group, we're going to have 2 for every single one of these individuals, which gives us a total of 600 big R's there. And so from this, we can calculate the allelic frequencies. We add up all of these numbers, which gives us 2,000 alleles total. And then we simply take the sum of these two and divide them by 2,000. So this for the little r's, you do 400 plus 500, and you get a total of 900 divided by 2,000, gives us 0.45 little r's. And then if we look at big R's, you do 500, 600, 1,100, and you get 0.55 big R's. All right, so 45% are have little r's and 55% have big R's. That's actually pretty close. You would think that a lot of times, especially when looking at the number of 200 smalls, that you would think that maybe that number was really not very prevalent. But if you look at just the alleles, it's actually not a whole lot off from the big R. So it's still fairly important as far as the population goes. And so you can also look at the changes here and see how that's changing over time. Is the big R becoming a larger number and the little r smaller number, or vice versa? And you can kind of see how the natural selection is occurring within that population by looking at simply the alleles. All right, so here's an example for you. I'm going to go quickly through this, basically just like we did the tulips, but a little bit quicker. All right, so your calculations looking at genotypes. So we're just going to be looking, we got green frogs and we got yellow frogs. All right, so for our genotypes, we have 25% big G's, 40% big G little g's, and we have 35% little g's. All right, so you can do your calculations there. For phenotypes, we actually have 65% green and 35% brown. And then for allelic frequencies, We have 45% big G and 55% little g. Right? So you can kind of look through these, and you'll see that the little g's, the recessive trait, is actually better suited for the environment. And so you can kind of follow through these, and again, over time, you might see these change, and you can start figuring out different things. But this is how we calculate out the genotypic frequencies, phenotypic frequencies, and allelic frequencies. All right. I'm going to skip through this a little bit. So we come to this idea of the Hardy-Weinberg principle. Right? And so with this Hardy-Weinberg principle, the idea is that the concept that a shuffling of genes, just simply through meiosis and sexual reproduction, cannot change the overall genetic makeup of a population. Since it's completely random and you got a 50-50 shot of within all those shuffling of genes, you're not going to get any evolution by just going through sexual reproduction. So just having that crossing over is not going to change a population's genetics. And so what does change a population's genetics? And so what the Hardy-Weinberg principle states is that as long as we maintain these 5 conditions that are met, a population will never evolve. So the idea is that these 5 things are what cause evolution. If you maintain and make sure that they never change, then you'll never have evolution. But in reality, you can never do that. And so basically, by their principle, they've been able to point out the 5 things that lead to evolution and natural selection. The first one is you have to have a very large population. So anytime you have a small population occurring in an area, like on an island, that can lead to evolution, right? So you must maintain a very large population. That can't always happen. You have to have that population isolated from all others. You can't get any kind of switching over. You can't have one organism leave one population and come to another one. They have to stay within their population, and that doesn't occur. You have immigration, you have migrations, you have all kinds of stuff going on. And so that's just not possible within nature. You can have no net mutations. Well, this is absolutely not possible. They've actually figured out the exact number of mutations that occur over time, and it's pretty much a consistency of always having these mutations occurring just because of mistakes that happen during mitosis and meiosis. Right? So you're always going to have mutations build up over time. Some of them are bad and will kill the organism. Some of them it doesn't matter and won't make any difference. And then some of them end up being adaptations. But there's no way within nature that you can have no mutations occurring. Right? So this is going to lead to evolution. Random mating. It doesn't happen. Organisms don't go eeny, meeny, miny, mo and just make babies. Right? They always have some kind of selective pressure, something that they're looking for. They want the biggest, the strongest, the best. Right? You have a great example are cardinals. Right? Male cardinals are bright red. The reason why they're bright red is because that color attracts everything, including the females, but also including predators. And if they're able to survive to adulthood being bright red and escaping all the predators that are always trying to kill them, they must have good genes. And so the brighter red that they are, the bigger and stronger that they are while being bright red, that shows how strong they are and how good genetics they must be. And so the females tend to pick those. So there is no random mating within nature. They always have some kind of selective pressure, something that helps them determine who they want to mate with. And then lastly, you cannot have natural selection occur. Right? Basically, you can't have any kind of competition whatsoever. And in nature, that just doesn't happen. You're always competing over something. You're always going to have some organism that's a little bit better suited for the environment than another, and so they're going to be able to reproduce more. So, but these 5 things, if you maintain all of these 5 things, you will never have any natural selection, never have any evolution. But as far as nature goes, that just doesn't happen. So that's why we have natural selection and evolution all the time. And so these are where we look when we want to figure out why natural selection is occurring. This is where we look to figure out that process. So we're going to go through each one of these and talk about it. So the idea here is that if all of those are met, it's at equilibrium and you get no change, no evolution. But that just doesn't happen. So the 5 mechanisms, we're taking one on one. First one, genetic drift. That whole idea of a gene pool of small population due to chance, right? So of having that very large population, if you maintain a very large population, then you're not going to have any changes. But that just doesn't happen. 2 examples here are bottleneck effect and founder effect. The bottleneck effect is when you have this very large population and some natural disaster comes along, smashes, kills everybody, and leaves behind just a very small group of organisms. Right? This happened to the human race actually a little bit over 100 to 200,000 years ago. Somewhere between 100 and 200,000 years ago, there was a massive volcanic eruption that basically covered the entire Earth with ash. And it caused basically this global winter. effect, killing off hundreds and thousands, they think up to possibly millions of humans on the planet. And it knocked down our population down to less than 1,000 people on the entire planet Earth. Right? That's a bottleneck effect. We went from this large population down to less than 1,000 people. We almost went extinct. Right? So that's this idea of a bottleneck effect. We have no idea what all those organisms, all those other people that died, what kind of traits that they have. We only know that the ones that survived are the ones that passed on their traits to us. So we know what traits they had, but we have no idea what kind of traits might have been out there and what kind of traits might have been lost. If you actually compare humans to each other, we are 99.9% identical to each other. We're actually closer, more identical to each other than chimpanzees are to each other, or pretty much most other species on the planet. Then you can take one organism to another and compare them genetically. We are much more identical than just about any other species. And that's because of this bottleneck effect that only happened about 100 to 200,000 years ago. The other idea with that is founder effect. Same idea, you have a large population on the mainland, and then you get this small population going out to, let's say, an island. And the great examples are Galapagos finches. Right? So you had this small group of finches that flew off from South America, got blown off, whatever hurricane, ended up on the Galapagos Islands, and you had this small group, this founder effect, this small group that started there, and they carried over those traits that they had. There might have been lots of other traits on the mainland, but the only ones available in that population were the ones that had gone over with that group. This also occurred in West Virginia. A good example is called the Blue People of West Virginia. If you Google it, you can kind of see a little story behind it. But basically, this group of people came over and colonized a small area of West Virginia. Being West Virginia, they were kind of locked within the mountains and didn't really get out a whole lot, and so they had a lot of interbreeding. And one of the families that came over into this town had a genetic defect that basically caused their skin to become very palish and blue, and especially when they were little, they literally looked like little Smurfs. And so they that population has, and this trait is only within like 1 in like 5 million people normally on the Earth, but within this small population in West Virginia, it's now become so popular it's like 1 in every 20 have it, and almost everybody's a carrier. So it's that trait is much more common within this small little town than it is within the entire world. And that's the idea, that changing of those genes because of certain events within populations. The next one is looking at, and so there's our ideas. So you got your bottleneck effect, the earthquakes, volcanoes causing populations to decrease. You have your founder effect, the genetic drift resulting in the colonization of a new group. It results in random changes of the gene pool because of this new population. So you got your Darwin finches, so you got the blue people. Mechanism number 2 is gene flow, right? So Hardy Weinberg said as long as you didn't have any, everybody stayed within that population, nobody left or nobody came in, then we'd be good. Well, this doesn't happen. You have lots of movement of individuals within populations. You have migrations, all kinds of different stuff. Step number 3, mutations. Change in an organism's DNA that creates a new allele. This happens all the time. Right? Non-random mating never occurs. Right? Or random mating never occurs. Non-random, they always have some kind of selection that they choose for their mate, whether it's biggest, strongest, brightest colors, whatever it is. There is always something that for choosing a mate. And lastly, that idea of natural selection. Right? This differential reproduction, the fact that some organisms reproduce more than others, is always going to lead to a change in that population over time. Right, so all of these help to show that you're going to get this microevolution, you're going to get these changes developing over time within a population. Now, modes of action. So how does this idea look when we start looking at populations and how they change over time? So we have 3 different kind of modes that we say of how this occurs. Normally, populations, when you're looking at them, have this bell curve, right? So you have, if we're looking at size, maybe you have some of them that are small, some of them that are large, but the large majority of organisms are in this middle size area, right? So this is your typical bell curve. Right, and so the number of individuals within the middle is much larger than any of the extremities. That are much more than the large or the small combined. Right, but when we start looking at when we start having this natural selection, it can change those. So you can have stabilizing selection, where basically the extremes are targeted. So anything that's too small or anything that's too large gets picked off, and the natural selection starts forcing everything to be that middle size, or and basically makes it stabilizing in weeds out those extremes. The best example of this is zebras. Right? All the zebras look exactly alike. If you get a big zebra, it's going to stand out within the group, and the lions are going to be able to pick it off. You get a small zebra, same idea. Right? The whole defense mechanism for zebras is that they look exactly alike, and it's hard to pick one out from the entire crowd. If one stands out for any reason, it's going to be able to be singled out and be killed by predators. So this creates a stabilizing selection within that zebra population. Okay? You have directional selection, which favors variance of one extreme. Right? So for some reason, the large ones are doing better. Maybe it's a new predator that comes in, whatever happens, but the larger of this population is doing better. And so the entire population starts to shift towards that larger group, to being larger and larger organisms. Right? And so we see this directional selection towards one of the extremes. Right? The key there is towards one of the extremes. Because we can also have the last type of selection called diversifying selection. And that means both extremes are selected for. So that means being really small is good, being really big is good, but being the average size is not good. All right? There can be a bunch of different things that can cause this, and this leads to creating two different groups or two different populations, and often leads to creating two different species. All right? So weeding out of these middle organisms and making the extremes actually better suited can actually lead to creating two different species this way, whereas the other ones can lead to having a different type of group. How fast does evolution actually occur? Well, Darwin's idea is known as gradualism. It's this gradual, slow change over time. All right, so you basically have these organisms slowly creating more offspring. The next generation is slightly better adapted. Then the next generation is even a little bit better adapted, and you get this slow change over millions and millions of years, thousands and thousands of generations. All right, that's the idea of gradualism proposed by Charles Darwin. But there's these two guys named Gould and Lewontin that came up with this idea of punctuated equilibrium. And with punctuated equilibrium, they noticed by looking at fossil records that we had this massive change in lots of different populations all at once within a very short, theoretically, geological time. And so by looking at fossils, they saw this big change, and so they came up with this idea of punctuated equilibrium. And what it does is it kind of coincides with mass extinctions. And so you'll have this mass extinction event, you'll have this big meteor come crashing down, kill off thousands of different species out there, and basically by killing off all those species, you still leave behind how they survived. So what's called their niche, their job in the environment. So what they ate, what they fed, how they survived, all those types of things are still there, but the species has been evaporated. The species is no longer doing that job. And so whatever's left behind can move into that species, or move into that job, move into that niche, and actually be able to survive within that niche and slowly kind of take over that job. And that causes a quick evolution to fill up that gap. And so whenever you have a mass extinction, you'll have this surge of new species coming in to fill in the gaps from all the other species that have went extinct. And this is the idea of punctuated equilibrium. And so the modern idea is that we basically have a combination of these two. We have the slow change over time, but whenever we have a mass extinction event, you have this huge punctuated equilibrium of all these different species all coming up at once. And so you get a massive change very, very quickly over generally short geologic time. And that's the general modern synthesis of the two. It's kind of a combination of both ideas. Now, we have four different evidences of evolution, right? We have fossils, we have amino acids and DNA, we have looking at their structures and their overall design, and we have looking at the embryology. So we'll go through each of these and then we'll kind of be done, all right? So a paleontologist is someone who studies those fossils. They look at structures and all those different types of things, what they ate, try to identify how they survived within their environment. And by looking at the fossil record, it basically provides evidence about the history of life on Earth. So we get to see kind of in order how it occurred, what organisms led to what, that kind of idea. And despite what you might have heard over millions from lots and lots of people, the fossil record for certain species is very, very clear and very, very distinct. Right? Whales are a great example. With whales, we pretty much have every single link of evolution going from when they lived on land to the modern day whales we have now. Every step, every adaptation, every mutation that occurred, every single species, we basically have an entire geological history of all the fossil evidence and all the changes that occurred throughout the entire process of evolution, going from when they were on land to when they became the whales that we think of today. So there is a complete fossil record for these guys. There are missing links as far as what's within our fossil record as far as where humans came from and lots of other species, but there are also lots of species where we have every single example found within the fossil record. especially things that tend to be marine or aquatic, because their bones tend to be covered very quickly and tend not to deteriorate, leaving fossils behind better. Right? So the idea is fossils are tough to make, and so certain environments provide better fossils, and those organisms that live in there we tend to have better evidence of. And more information for it. Right? So this fossil record is not like one of these things of, they don't really know what they're talking about. They actually have, for whales, every single step along the evolutionary process. All right, so this is not just something that people are kind of saying, well, it kind of fits in. They are very the fossil record for a lot of species is very distinct and very thorough. Now, and this is the idea. You're starting off with very, very early time, going all the way up until you finally hit to today's time, so you can kind of see the geological scale, how many millions and millions of years goes within those, and you can also see the rock layer. So the oldest ones are always going to be at the bottom. The newest organisms are always going to be at the top of the rock layers. So either the remains of an organism or some trace of the organism is what makes a fossil. All right, so you're going to have the bones left behind, an imprint left behind, something along those lines. So not every organism that dies will form a fossil. Actually, very few of them do. All right, so it's very difficult for fossils to get made. Most of the time, the organisms get eaten, get decayed. Something happens, and those fossils don't develop. You have to have very specific occurrences to happen in order for a fossil to occur. All right, and so it's actually very difficult to get fossils, which is why we don't have that many of them in reference to how many organisms have died over the millions of years. So typically water carries small rock particles to lakes and seas. Dead organisms are buried by all that sediment and preserve those over time. So you can kind of see a nice little picture here. You can have where it actually creates a fossil this way, or where it creates a fossil and actually gives us a shape. And we have this term called relative dating. Right? Relative dating is not dating your cousin. Right? Relative dating is looking at how things stack up when we're looking at the entire fossil record. Right? You're going to have to do this on the quiz and exam that you're going to have. Right? And so basically you need to look and you need to compare layers and you need to see which layer is the oldest. Right? The oldest is always at the bottom. You're going to have you can have 2 or 3 different sections, and you're going to have to look and see which ones match up and how they match up. And you're going to have to figure out what layer is the oldest. So within this, looking at this one little diagram, the oldest fossil is the trilobites down here at the bottom. Right? They're well below these clamshells up here, and so these are going to be the older species, and these will be the newer species. The newer ones get added on top as we go through. So you always, whenever we have a question involving how old something is, you always want to look and try to find the bottom layer to find the oldest fossil. We also can have radioactive dating. So this is your carbon dating and all that kind of good stuff. All right. So they look at the radioactive material within the specimens that they're looking for, and based on how much of that radioactive material there is left, based on how much was there when it got set in, they can calculate how old that organism is. All right. Certain radioactive isotopes are better for certain times. All right. Carbon is good for 10,000, 20,000, 30,000 years old. But when we're talking about a million years, carbon doesn't do any good. Once you get so much, you basically can't detect it anymore. So that's when they move on to other things like iodine, plutonium, lead, all kinds of different ones. All right. So certain isotopes are good for certain ranges as far as figuring out times. When we start looking at amino acid sequences as far as figuring out how organisms are related and how evolution occurs, we're looking at their DNA. Right? So the greater similarities in their amino acids, the greater similarities in their DNA, then the closer the relationship. The more differences you have, the longer that they've been separated and the more distant the relationship is. So this is basically one of the most recent types of trying to categorize organisms. And then the most ancient one, or one of the first ones, is looking at their structures. So we start looking at different organisms alive today. We look at their the structures that they have, and we try to determine which ones are closest related by which ones have the most similar structures within them. We have these 3 terms that you got to know for structures. You have homologous structures, analogous structures, and vestigial structures. Vestigial means used to be around or used to be important, but now it's kind of just wasting away. Kind of like your tailbone, your pinky toes, your appendix, sort of, that kind of thing. So it was a structure that used to have some importance, but now is greatly reduced importance. Homologous means that there are organisms that have similar structure, or that have similar parts, but look very different. They come from a common ancestor, so they're going to have similar structures within their bodies, but those bodies might look very different on the outside. Analogous structures are ones that they happen to look very similar, but they're actually not closely related. Right, so this diagram is basically a great example of homologous structures. You have the human arm, you have the leg of a horse, leg of a cat, wing of a bat, wing of a bird, and the fin of a whale. Right, all of them look very, very different when you look at each of the organisms. But if you look within those structures, they all have a humerus. They all have the ulna and a radius within their structures. They all have these phalange structures, right? So they all have these basic designs and these same basic structures within every single one of them. Some of the bones might be a little bit longer, some of them might be a little bit shorter, that kind of idea, but they still have those same basic structures within all of them, even though they look very, very different. And that's because they all come from a common ancestor that had those basic structures. Right? And so over time, those things have evolved and changed what they look like on the outside, but they still kept a lot of those basic internal structures the same, at least it's the same different basic design. Okay. Analogous is where they basically are not closely related at all, but they live in similar environments. So you got with fish, dolphins, sharks, all those types of things, right? So sharks and bony fish and dolphins are not very closely related at all, but they all have fins. They all have the same basic shape. They all have very smooth body contours. They all have tail fins. All those types of things, right? Because those are what are best suited for living in the water and moving quickly, right? So they allow the organisms to turn very fast. They allow to generate a lot of push. They allow them to not be very hydrodynamic and not use a lot of energy. All those types of things within that population help them do better. And so, since even though they're not closely related, they look very similar as far as their structures. Same idea here with all these organisms that fly. Right, you have these large wings with lots and lots of surface area who have similar kind of bony structures within them to provide them rigid support. All those types of things and to help basically create lift and create power and lots of surface area. Right? They're not closely related at all. One of them's a reptile, one of them's an insect, one of them's a bird, and one of them's a mammal. So they're not very closely related at all, but they all have very similar structures because they live in the same environment and the same natural selection pressures are applying to each one. And so whatever works well for one is going to work well for all of them. And the last kind of idea for a proof or support for evolution is the idea of embryology. This is one that I tend not to use. The other three are so good that this is kind of a little wishy-washy for me. But it is one of those evidences. So what they say is that when you're looking at the embryology, when you're looking at how organisms develop, you're going to have them when they're very early within their development, like all of these guys up here. They look very, very similar. So even though you have a lizard, tortoise, pig, and human, they all look very, very similar, like little tadpoles, basically. And then as they start to evolve, is when they start to develop within the womb or within the egg. They start to kind of separate out. And so very early on, we all have the same basic characteristics. And then as you move along through our development, you also kind of move along through our evolutionary steps. So the lizards and the reptiles all start looking very, very similar to each other, but the mammals start to look very different from those. And so you'll have these groups, and they slowly start looking more and more different, the more and more advanced they become. All right, so that's the idea of embryology. And the last kind of thing to talk about is how life developed on Earth. So there's a bunch of different theories. The first one was abiogenesis. Basically, life just kind of sprang up out of anything, right? And so the idea with abiogenesis is that life kind of pop out of anything, and when it was kind of disproven by these guys named Redi and Pasteur. And what Redi and Pasteur did is they created experiments here. Redi did his experiment with a glass jar and some meat. Pasteur did his experiment with some chicken broth in this little glass tube. And basically what Redi did is he put some meat in there, put some cheesecloth over it, put some meat inside of this jar, no cheesecloth, and his experiment basically proved that the one that had no cheesecloth on it, the flies were able to get to it and ended up having maggots. The one that had the cheesecloth over top, the flies couldn't lay their eggs on it, and therefore there were no maggots on the meat. And so he proved that the maggots came from the flies, not just kind of popping out of the meat. Because at this point in time, people actually thought that maggots just naturally came from meat. And so they didn't do anything to cover it or keep any insects off of it. They just kind of left it sitting out all the time. Because they thought that the maggots will just come from the meat no matter what they did. And what he proved is that the maggots actually came from the flies. Pasteur used this cool little glass jar. He put some chicken broth in it. He put some chicken broth in a normal jar, and the chicken broth in the normal jar ended up getting all this mold and nasty stuff in it. But this chicken broth in this special jar ended up not getting mold or anything in it, and that's because all the mold particles got trapped in here. So he proved that all the mold and the things that made the chicken broth go bad was actually found within these dust particles here. And so if you ever heard Pasteur, you got pasteurization. And the whole idea of started preserving food. If you can it up, or if you put it in a sealed container, you kill off all the bacteria and stuff within it, it'll stay fresh much, much longer. So by both of these studies help to improve food preparation over a long period of time. All right, so that's our evolution for this time. So take a look over those ideas, think about what we've talked over, watch this presentation over again, and be prepared for class. Thank you very much. Bye-bye.